Welcome everyone to today's first seminar from the semester. It's a great pleasure to welcome Dr. Jeffrey Shaman from Columbia University from their School of Public Health. Jeffrey Shaman did his bachelor's degree in biology at University of Pennsylvania. Then he came to Columbia University, to IRI actually, for his master's and PhD work, where he did his research with Mark Kane. And his doctoral thesis was actually about the impact of hydrology on the spread of West Nile virus. After his, and he was awarded a prize for exceptional work as a graduate student for his scientific research at Columbia. Then he was a postdoctoral fellow at Harvard University, where he worked with Dr. Zipperman. And then he became an assistant professor of, at Oregon State University in Corvallis, and then transferred to Columbia, where he's a faculty member. So Jeffrey Shaman has worked on classical climate problems, and he has many publications on climate change, on monsoon dynamics, on African climate, and so on. But he has also worked on the interface of weather and climate and disease. And so one aspect of it he will present to us today. So let us welcome Jeffrey Shaman.
If it's better for me to play slower. Thank you for having me here. It's a pleasure to come. So I'm going to be talking today about some of my work at the interface between climate and meteorology and disease, and I'm going to specifically be talking about influenza. I'm going to be building to talk about pandemic influenza, and obviously we had a pandemic recently in 2009. But before I do that, I want to talk about some of the other work we've done that looks at seasonal flu. Okay, and the distinction between seasonal flu and pandemic flu I'll delineate a little later in the talk. But first I want to talk about just the seasonality of influenza, which is sort of its climatological characteristics, if you will. So the plot here shows you average daily excess pneumonia and influenza mortality for 31 years in 2 states in the US, California and New York. It goes from January to December. And what it shows you is something that most of us intuitively know, particularly if we've spent any time in temperate regions in the world, and that's that in temperate regions of the world, the flu occurs in the wintertime. And this is something that's been observed for over 2,500 years. And the reason for it is not really well known. There are ideas about it, and you can sort of group those hypotheses into 3 basic groupings. The first has to do with changes in host behavior. So the idea there is that people in wintertime spend more time indoors, they're more crowded with each other, amongst each other. Children are in school, and that increased contact, that increased person-to-person contact facilitates the transmission of influenza, and that results in this bump in the number of cases and this rise that we get. The second grouping has to do with host-mediated factors. So these are changes in the actual physiology of humans, and in particular changes in our immune function. So the idea there is that there's less light, day length is shorter during the winter time, the sun is at a lower angle in the sky, there's less vitamin D, less melatonin production in people, and that actually suppresses immune function, and that suppression of immune function is enough to result in greater infections, greater rates of viral shedding for those who are infected, and as a consequence, greater transmission in this wintertime peak that we see there. The third has to do with environmental factors purely. Things like temperature and humidity, which actually may be modulating the survival or the dynamics of the virus itself once it's expelled into the environment. So those are sort of the 3 groupings of ideas that go behind this, and I'm going to talk a little bit about some of the work that I've been doing with colleagues to look at the seasonality of influenza for this recurrent flu structure. Now, before I do that, I should say that there's a lot we don't know about flu, and some of it is actually almost embarrassing to admit. So for instance, we don't know how flu is transmitted. I mean, it's something that everybody in this room, I'm sure, has had, but we actually don't know how it's transmission. We don't know how it can be transmitted. We know theoretically there are basically 4 modes by which humans can pass it from person to person, but nobody's observed flu being transmitted, so we don't know what the dominant mode is. So the 4 modes are direct contact. So if I'm sick with the flu, I'm shedding it, it's in my mouth, my nasal cavity, I kiss my wife, it's direct contact with her, I'm going to be giving her the flu, okay? The second is indirect contact, and that has to do with objects which we call fomites in business, I guess, and a fomite is any object upon which you might deposit flu, okay? So if you're sick with the flu, and you may even be asymptomatic and sick with the flu, you'll be shedding the virus. It comes out as you speak, it comes out as you cough, it comes out as you sneeze. That's a picture of a human sneeze, by the way. Most of it comes out your mouth, that's what you want to cover. It comes out even as you're breathing tidally, okay? And when you do that, you have a whole array of droplets that are coming out, and some of them are going to settle on objects. So if I'm sick with the flu and I come up here and I speak and I'm depositing droplets, some of which may have the flu virus in them, all over the podium right here. And the next person who comes along, audio video guy, Mark, he seemed really nice, he comes up here, he touches it all there, and then he rubs his eyes, or if he picks his nose or something like that, he's going to be transferring to himself. That's the indirect route. Droplet is the third, and droplet's sort of almost like direct. Sometimes people couple it into direct, but droplet is that spray column right here. If I'm talking to somebody, I'm spraying directly in their face. Whether you know it or not, people, when they speak to each other, spray each other. So if that spray contains viral particles within those droplets, you're going to potentially infect somebody. The fourth is airborne. So the idea with airborne is, again, you have this distribution of droplet sizes that's expelled, some of which for a sick person are going to contain viral particles in them, okay? Some of them are going to be very large and settle out. Some of them are going to be very small, and they're going to be small enough that they're going to be aerosolized. They're going to remain aloft on the turbulence in the room, and they're going to float about the room for hours on end, in which everybody's breathing here, presumably. You can inhale it. It can get into the lining of your mouth, lining of your nose, into your lungs, and you can get infected that way. So these are the four modes of transmission, and it was thought through the 20th century until about the 70s that the predominant route was airborne. Then in the 70s, they started to think, well, maybe fomites have something to do with this, and are very important. That's why they're always telling you to wash hands. The evidence that washing hands actually helps with influenza is actually not that strong. There's some, but it's really not that strong. Diarrheal disease, washing hands is very key. But flu, it's not really clear that it's a strong effect. Right now, basically, people just argue about it. In the literature and in person, it's kind of funny to watch. I think the pendulum is swinging back towards airborne, and I actually personally think airborne's probably the more dominant route, but it's really hard to know, and it may vary from place to place. So that's some of the confusion that is sort of the background about flu, and I'll give you some more. And this brings me to this experiment that was performed by Peter Palaze's group. He's a virologist at Mount Sinai Hospital in New York, and they did an experiment with guinea pigs. They found that a guinea pig was a very good animal model for studying the transmission of flu. And what they would do is they would set up a chamber that looked something like this cartoon that they created themselves, in which they would take 8 guinea pigs, they would infect 4 of them with human influenza, and they put them in separate cages right there. Next to them, stacked in this vertical array, they put 4 susceptible pigs, those that were going to be exposed. They then put it all in a chamber like this, with a slight airflow going from infected to exposed, and they'd set the chamber at a fixed temperature and relative humidity condition for 72 hours. They'd monitor the shedding rates of the infected guinea pigs, and they'd determine how many of the exposed guinea pigs, 0, 1, 2, 3, or 4, after 72 hours became infected. They ran this 20 times using different temperature and relative humidity conditions. What they found were some marginally statistically significant effects in that colder temperatures and lower relative humidities favored the transmission of flu amongst the guinea pigs. So I was reading this, and my background is more meteorology, and I was asking myself, well, why are they using relative humidity? Okay, obviously we hear about relative humidity on the local weather report, but it's not really a constrained variable in the sense that you can change relative humidity either by changing the humidity content of a parcel of air or by just changing its temperature. So this is the equation that shows you it in terms of vapor pressure. V is the vapor pressure, and most of you may know this. V is the vapor pressure of the air, and E sub S of T is the saturation vapor pressure, which is the point at which condensation and evaporation rates equilibrate or equal. And it's just that ratio that determines relative humidity. And the thing about the saturation vapor pressure is that point at which you hit 100% relative humidity, the point of saturation, the point at which fog forms and a cloud begins to form. varies very strongly as a function of temperature. So again, if I want to change the relative humidity in this room, and I close it, I can do it two ways. I can set up a dehumidifier or a humidifier and change the actual water vapor content of the air, or I can just monkey around with a thermostat. I can change the temperature in the room, and that will change the relative humidity. So the question is, well, is this really that important? And the argument is, well, yeah, it is. It's not really an argument, it is. So the fact is, it's the Clausius-Clapeyron relationship, which is this non-linear exponential relationship between temperature and absolute humidity, here expressed as this term vapor pressure. And as you can see, it is in fact exponential. And this is the line for 100% relative humidity. Here's the line for 50% relative humidity. And those two x's mark very real conditions that you experience right here between summer and winter. One's 5 degrees Celsius, the other's 25 degrees Celsius. Both have 50% relative humidity, but the warmer temperature has almost 5 times as much water vapor in the air as the colder temperature. So the question is, well, why not use a measure of absolute humidity? Instead of using relative humidity, why don't we just take a look at absolute humidity? So that's what we did. We just culled their data, used the Clausius-Clapeyron relationship here, and just backed it out. And we redid their analysis just using some regression. And what this shows you is the regression of their guinea pig data on relative humidity, temperature, and vapor pressure as backed out from these two variables, which they had set in their experimental chambers. What we find is what they found before for relative humidity and temperature, which are these marginally statistically significant effects such that you're getting more transmission, more of the guinea pigs are becoming infected as you go to colder temperatures or you go to lower relative humidities. Then if we do it with the vapor pressure, we find actually a much more statistically significant effect. So we saw this and said, okay, well that's interesting. Subsequent to that, they published some more data that was all performed at high temperatures. So basically they got a huge cluster of points when they added the additional data right here. It sort of almost biased the data on the temperature because they all got zero transmission at high temperatures. And what they found there, what we found there when we added this in is that it sort of broke down the relative humidity a bit. It strengthened the temperature, again, because of the redundancy right there. And we start to see for the absolute humidity, a bit of a curvature, almost a nonlinear relationship perhaps there when we have more data because it actually spreads out and adds a variety of points to the absolute humidity conditions. So we're still getting the same relation, and it's still very strong for absolute humidity. Well, the authors of the study, Lowen et al., this group at Mount Sinai, had put forth two hypotheses to explain why relative humidity might be associated with the changes in the viral transmission amongst the guinea pigs. The first was that the virus-laden aerosols, so again, you're spewing out aerosols. The guinea pigs don't sneeze or cough, but they do breathe, obviously, and they chatter, and so they're expelling droplets. And they can't touch each other, by the way, in this chamber experiment. So this is an airborne transmission route that's going on right now. So the virus-laden aerosols, which are also referred to sometimes as droplet nuclei, are more efficiently produced at lower relative humidities due to increased evaporation of the expelled droplets, such that more virus remains airborne longer. And what that's saying again is that if you have a distribution of droplet sizes expelled, some of which are going to be large and fall out of the air and settle on something, some of which are going to be very small and remain aerosolized on turbulence, you also have a mid-range section, okay? And those are going to be governed by two competing forces. One is the rate at which they fall, and the other is going to be the rate at which they evaporate. And if they evaporate before they actually hit the ground, then in fact those particles are also going to join that pool of particles that are aerosolized. So the idea is that if there's more evaporation going on, if you're increasing the rates of evaporation, you're increasing the rates at which you have aerosolized droplets, and you're going to have more virus floating around, and that's going to increase the transmission potential, okay? So we first took a look at this. So basically we said is, well, why don't we just model it? Let's take an ideal model. We'll just use a droplet of water. We know that's ideal because obviously expelled droplets are not just water, but let's look at a droplet of pure water, and let's compare the two rates of sedimentation and evaporation, okay? Just the two equations for it. Sedimentation rate, at least the terminal velocity, is going to be related to how large the droplet size is because there's Stokes drag force acting in the opposite direction and there's gravity moving this way, reaches terminal velocity that's dependent on the radius of the particle right here, that r squared right there. And then the evaporation rate is going to be this thing right here, which is a function of vapor pressure, saturation vapor pressure, this difference right here, which is the vapor pressure deficit, temperature, radius, and some thermodynamic variables. So what we can do is we can hold a droplet a meter above the ground. We can set the conditions in the model as wherever we want, let it go, and see whether or not it hits the ground, how long it takes to hit the ground, or if it evaporates to the point where it's small enough where we're going to say, all right, it's aerosolized, okay? So this somewhat peculiar plot shows this right here. The colors actually show you how long it takes for the droplet to hit the ground. In this area right here where it goes white, the droplets actually became small enough, 2 microns in size, that we said, okay, they're aerosolized. And what we found essentially is that the droplets do aerosolize more, and you are going to get more droplets, but it's going to be actually at higher temperatures and lower humidities, not colder temperatures and lower humidities. So it's a little bit at odds with what they found, and consistent with what I'm sure some of you were actually intuiting. We then said, okay, well, look, really what they're talking about here is evaporation. If you want to get at evaporation, you're not really talking about absolute humidity, you're not talking about relative humidity, you're really talking about the vapor pressure deficit, which is, this is this evaporation equation, the rate at which the radius of the droplet starts to fall away, and it's really dependent on this guy right here, the vapor pressure deficit divided by temperature. What we're saying then is that if that's the force that's driving it, this is the thing, not relative humidity, not temperature, not absolute humidity, that should pop out from the data. That's the thing that really should be best associated with that transmission data if evaporation dynamics are what are controlling the rates of transmission. Well, that's the regression. Doesn't give you anything. It's pretty garbagey, right? So it doesn't seem like that hypothesis really holds pretty well. Well, they had another hypothesis. And the second hypothesis is that influenza virus survival increases as humidity decreases, such that the airborne virus remains viable longer at lower humidities, okay? And what this actually calls upon is studies that go all the way back to the late 1930s, where people would actually culture influenza virus, they'd atomize it inside a canister that looks something like this. It's called a Goldberg drum. Just a rotating canister that they would set at fixed temperature and relative humidity. They always looked at relative humidity conditions. And then what they do is they go in and they'd sample the air and see how much of the virus had remained viable through time. And you could see just how it decayed away, whether it increased over in certain conditions or decreased the rates of decay or survival of the virus. So we went looking through the data, and there are a number of studies, as I said, all going all the way back to the late 30s that do this. And we found really one study that met 2 criteria that we really needed to do this reanalysis of it. First is that it tested a range of both temperature and relative humidity conditions. And the second is that it actually had the data in the paper so we could actually play with it, because a lot of them were old and there was no data published in the paper. So this was this data of Harper from 1961. And what this shows you on the left are scatter plots from relative humidity going from low to high, temperature from low to high, and again, vapor pressure as we calculated it out. The red is survival at 1 hour, green is survival at 6 hours, and the blue is survival at 23 hours. What you can see is the scatter. Again, there's more survival as you go to lower relative humidities. There's more survival as you go to lower temperatures. And then when you go to absolute humidity, it's looking pretty tight, right? As a matter of fact, if you just do a log regression of this to take care of that exponential in it, 90% of the 1 hour survival is explained by absolute humidity by itself. So we thought that that was a pretty good, if not smoking gun, at least very, very encouraging. So the other thing that's important when we go back to thinking about the seasonality of it, what we're finding is that low absolute humidity conditions seem to favor both the survival and the transmission of the virus in these laboratory settings, right? Well, if we look at what's going on seasonally in temperate regions, here's some indoor climate data taken from Scandinavia. The blue is absolute humidity, measured as vapor pressure. Here is outdoor vapor pressure taken from NCEP-NCAR reanalysis, basically, and it's just 30 to 60 degrees north. And you can see both of them for these regions have these very pronounced seasonal cycle that's in phase with what you'd expect. That is, it's a low in wintertime, and it's the low absolute humidity that seems to favor the survival and transmission of the virus. Relative humidity has a problem, though. Firstly, it's differently phased indoors to outdoors. So in the indoor environment, it's correctly phased, which is actually pretty good because we spend about 90% of our time indoors. But in the outdoor environment, it's actually the wrong way. Furthermore, it doesn't actually range over a huge range of values on average, though there's differences from building to building. not actually very well characterized what indoor humidity conditions look like, so there's a limited amount of data, but this is consistent with what you generally find. So it's not correct for the outdoor environment, and the statistical relationship is a lot weaker. Furthermore, if we were to think about temperature, temperature is very highly managed indoors in most temperate regions of the world, and people spend 90% of their time indoors again, and they don't experience a huge fluctuation in temperature. So unless you're going to call upon people having a lot of the transmission taking place outdoors, it doesn't really make sense. So bottom line is that the laboratory data supports the fact that absolute humidity seems to have a stronger association with the survival of influenza virus, stronger association with the transmission of the virus, and this is consistent with the seasonal changes both indoor and outdoors that we see in absolute humidity. They're appropriately phased so that you would expect it to modulate influenza transmission rates as observed. So, questions that then fall from this is, well, can we scale this up? Can we take this laboratory findings that we have, and can we put it into a model and actually model what's going on, let's say in the United States, and see if we can match it to epidemiological data? Can we match it to something at that scale? In addition, is it associated with changes in influenza transmission dynamics? If you take the model aside, are you just going to see some sort of statistical relationship between absolute humidity and influenza transmission? And then another one, which is still an outstanding question, is, well, what's the mechanism by which absolute humidity affects influenza survival? Why does a droplet expelled in water and glycoproteins and broken cell particles care about ambient absolute humidity? We don't know the answer to that as of yet. So to model it, we said, all right, well, let's mimic this relationship here. And I'm going to switch from vapor pressure to specific humidity because it's just easier to get absolute humidity and specific humidity data. Very similar relationship. Here's the influenza virus transmission, regressed on specific humidity. There's that Lowen and all Peter Palese data. And you can see, again, it's got sort of this nonlinear fit right there, same with the survival data. So basically, we're going to have some sort of functional form in our model that translates observed local daily absolute humidities, in this case specific humidity conditions, into the force of transmission in the model, which is called R0 in the epidemiological literature. So R0 is the basic reproductive number. It's how many infections an infected person would cause in a fully susceptible population. So if there's an R0 of 2, and there are 10 people infected, they're going to infect 20 people, those 20 will infect 40 people, et cetera. It's a growing epidemic. If it's an R0 of 5, those 10 people, or 0.5, the R0 of 0.5, 10 people will infect 5, and then 2, and then 1, and the epidemic will die out. Okay? So to measure the force of transmission. So what we've just done is we've just made this funny equation which uses specific humidity, which is here Q of T, to give us some variant of R0 of T. There are two parameters that determine the range over which it varies, okay? R0 max and R0 min, and we're going to select those through the model experiments. We're not going to sign them up right away. And this is what we use to model it. It's a very simple and standard epidemiological model. It's called an SIRS model, which stands for susceptible, infected, recovered, susceptible. And what it does basically is it divides the human population in this perfectly mixed ideal context into 3 classes: those who are susceptible to becoming infected with the pathogen in question, in this case influenza, those who are actually infected with it, and those who have been infected with it but are recovered. The second S, the last one on the end there, basically indicates that, as with flu, you can get infected more than once. After you've recovered, at some point of time, you're going to lose your immunity to that prior infection. And this has to do with the fact that there are multiple subtypes of flu floating around, and that they're always changing, and that's why people can recurrently get the flu. So basically, it's just these two ODEs coupled to each other. It's just a nonlinear oscillator, and it has 4 parameters in it. L is this duration of immunity, which is that time going from that R category to S, how long you stay immune. D is the mean infection period, how long a person on average stays in the I category once they're infected. And then R0 max and R0 min, which just determine that functional relationship between absolute humidity and this force of transmission, and which then factors into this beta term there, along with D, which is in there and determines the dynamics of the model. And when we ran it with 2 subtypes. We actually put more than 1 flu strain into the model, H3N2 and H1N1/B. So basically, we seeded a model over the course of a 31-year run with what was the dominant subtype of record for that model. We ran it retrospectively. We had some stochasticity in it in the sense that we're always seeding the population because we're running it locally, and flu is very often imported. It moves around the planet, goes from the northern hemisphere to the southern hemisphere out of year, and then it's usually year-round in the tropics. And then we assessed the fit of the model compared to excess weekly pneumonia and influenza mortality, which was that seasonality cycle I showed you in that very first slide for California and New York. Okay? So this is what it looks like. All right, we had 5,000 model simulations, and we did it with a range of the model parameters here. It was taken from the literature, and we broadened it a little to be inclusive. We would take a record of New York State specific humidity. There you go, that's a daily record for 31 years. We did this for 5 states, Arizona, Florida, Illinois, New York, and Washington. Have some varied climates in that sense, so that's why we picked those different states. We do 5,000 simulations, each run with a different set of parameter combinations, okay, different parameter combination. And this is what the output would look like. We have some sort of simulated rates of infection popping along. What we're interested in is whether or not we can model the seasonal cycle. Are we going to get the actual seasonal cycle? And on the left here just shows you the seasonal cycles of daily specific humidity. You can see just how they're different for the different states. And here's the output for the 5 states. And what you can see in the blue line is the best fit based on those 5,000 simulations. The red line is the observed excess pneumonia and influenza mortality. The green is the 2nd through 10th best fits. And what you can see is we're capturing some of the basic features, which is this very late season rise that's taking place right here. Sometimes the double peak that you see here that has to do with different timing preferences for when the peak is going to occur in different years. And that's great. It actually fits all 5 of them well. But more importantly, the best fit simulations for each of these states are drawing upon the same parameters, okay? So if we needed, the parameters in here are very important because they reflect the biology of the virus, okay? And if we needed one set of parameters to make it work in New York and another set to make it work in Arizona and another set to make it work in Florida, we're in essence saying, I need a different virus in each of these places in order to make this work. And that's going to break down the whole idea. But that's not what happened. In fact, we found that they were converging to the same parameter space of a very high R0 max term, a high R0 min, and a low mean infectious period, short duration of actual infectivity, okay? So this was very good. This actually said that they were all acting in consort with each other. And then what we actually did is we then cross-validated it. We took these fits, the best fits from these 5 states, and we applied it to the rest of the country. And when we did that, we were able to get really good fits for the rest of the countries, except for a few states where the data was actually very data poor. And those were the ones that didn't work so well, and we kind of expected them not to. So basically what we were able to see with this model is we were able to describe the seasonal cycle with just this one variable in the model with some degree of fidelity. We then said, all right, well, let's just take the model and just put it aside for the moment. Can we see some direct imprint, some direct legacy of absolute humidity in the raw epidemiological data? So to do that, we did sort of a, I call it an anomaly analysis, I guess. And so what we did is we took each year. As a matter of fact, I'm going to cut way ahead here. Then I have a plot here that's stuck in here that'll make it a little clearer. That's it right there. And what we did is we went into each winter at each location. Here's Florida for 4 winters within that excess pneumonia and influenza mortality record. And we said, let's find the period at which mortality for 2 weeks straight rises above some threshold level. We'll vary the threshold just to make sure it's not sensitive to it. And we'll call that the onset, okay? The start of the outbreak. So here are 4 levels shown, and you can see here you've got 2 peaks and it's kind of squiggly coming up, but this is the first period, 2-week period right here, where mortality continuously exceeds that threshold. Here, it's kind of a dud. It doesn't work too well for this winter. Here's a very nice single mode peak you got there, and then all 4 are clustered right there. And then here's another one with a much later, longer peak that 3 of them capture right there. So anyhow, we label that onset for each of those winters in all 48 of the contiguous US states plus the District of Columbia, and we call those onsets for each of those 30 winters, okay? We then look at what did the humidity conditions look like prior. Now we're comparing onsets that take place in different locations and at different times of year. So we can't just look at the humidity data, we have to look at anomalies of humidity. We already know it's the winter time. We already know that conditions locally are at their seasonal low. But we want to know if they're anomalously low. Is this occurring when things are even drier than they typically are, or more humid than they typically are at that wintertime low. So because of that, we look at the anomalies, and that allows us to analyze across all these different times and different locations. And I apologize for having to run through this backwards and forwards to your dizziness. All right, so this is what we found when we did it. So we've lined everything up to that day relative to the onset. And this is for combinations anywhere from like 1,200 to 1,400, depending on the threshold of different anomalies, different onset dates, averaged. And what you can see is that for the 3 to 4 week period prior to the onset, there's a big drop in the humidity on average. Okay? It's not huge, mind you. I mean, this is -3 times 10 to the -4th, but it's actually significant when you put it in terms of what absolute humidity or specific humidity would be, and it's quite statistically significant. Now, it's not happening all the time, okay? You don't need to have this anomaly in order to have an outbreak start to foment. In fact, it only happens in 55 to 60% of the cases that you actually have a negative anomaly at all. But that's still much more likely than when you would have otherwise. I mean, what it's really saying is that negative humidity anomalies, and remember, these are anomalies below what's already the low wintertime humidity conditions, are not necessary for the onset, but they presage an increased likelihood of actually having an outbreak. Basically, when you have those negative humidity anomalies, you're boosting the virus survival above what its already typical low wintertime levels are. So if the virus is there and it's circulating and susceptibility is appropriate, a bunch of other conditions, some of them biological, it's going to actually encourage that outbreak to take place then and there. So we see this actually in the data, and it's a pretty nice indicator. And one other thing we did is we went back and we said, well, let's go look in the model and see whether or not the model behaves the same way. And in fact, it does. The model only has humidity going on, so it's actually a much cleaner signal. But we also see this in the model as well. There's not all the other dynamics that are potentially going on as you see in the real world. All right, so absolute humidity scales up to provide a single coherent explanation for the observed variability of influenza seasonality in temperate regions. At least that's what this analysis and modeling from the United States would suggest. Next question is, well, can we associate this laboratory and epidemiological finding, which is really a local effect, right, with larger climate phenomena, something that takes place over broader timescales and in particular broader regions? So let's bring this up to ENSO. So ENSO, obviously, a lot of you know about this. Study it is one of the more studied climate phenomena around, and after the seasonal cycle, it's a thing for which we may have the most predictive skill. It's a coupled ocean atmosphere phenomenon that takes place in the tropical Pacific, expands the whole 12,000 plus kilometers of the tropical Pacific, and it basically, in its neutral state, its average state, it's characterized by easterly trade winds, move from east to west over the water, and create wind stress on that water, and essentially push the water and pilot it up in the western Pacific. Creates a big warm pool there, a pool of warm water, and it pushes down the thermocline. On the other end, as the water is being pulled away over here, you get a divergence and ethnic pumping, and that shoals the thermocline up there. So you get colder sea surface temperatures in the east and lower sea surface heights. Now these warm sea surface temperatures right here support a lot of convection. The cold sea surface temperatures here do not. So what you tend to get is a lot of rising motion here that then circulates over in a Walker circulation and subsides over here. So you have this reinforcing phenomenon where the sea surface temperatures are supporting the convection that's supporting the trade winds that's supporting the sea surface temperatures, okay? And there are two phases that are extremes in the ENSO system, La Nina and El Nino. La Nina is the one where you have increased trade winds right here, an even larger, warmer pool over here, and colder temperatures here. And El Nino is the opposite, when you have a relaxation of the winds and the water starts sloshing back across the equator, and you get a warming on the eastern side, and you get convection anomalously there. Now, this has been studied a lot. We have some predictive skill of it. We don't fully understand the dynamics. One of the things we do know about it is it's a big area, huge expanse of water. Well, it has a lot of what are known as teleconnections, which are these statistical associations between what's going on in the tropical Pacific and weather events elsewhere. A lot of these are mediated by the atmosphere, not all of them, but basically ENSO events, changes in the phase, are associated with changes in temperature and precipitation and humidity around the planet. So this is just a map showing you some of the changes associated with El Nino in boreal winter and then boreal summer right here. And then here's another map over here showing you the same thing but for the La Nina phase. And you can see they're somewhat symmetric, but they're not completely symmetric, okay? So we have these teleconnections that take place, and in addition to affecting meteorological conditions at the regional scale, you could argue, given the size of those anomalies, this meteorological variability has been shown to affect and documented to affect a lot of other sectors. These include things like agriculture, economic productivity, fisheries, ecosystem health, and human health, okay? And because ENSO is predictable, robust relationships between ENSO and these other sectors can allow prediction of those outcomes. So there's something attractive to being able to find an association with ENSO, have some predictability of ENSO, and therefore have some predictability of that outcome itself. So what are some of these things? Well, one of the earliest examples was done by my former advisor, Mark Kane, and when he looked at maize yields in Zimbabwe, Southern Africa. If you remember right here, I definitely remember right here, there's a big connection, a big teleconnection in Southern Africa, where Zimbabwe sits. And you can see there's a very high correlation between an index of ENSO variability and maize yield. So you have this ability to predict what's going on potentially there, and actually adjust your plantings accordingly based on those predictions. Here's another one. ENSO has also been shown to affect the health and behavior of migratory birds by altering their biomass, their flight and stopover patterns, their time to molt, and the population densities, okay? And here's the third. People have actually looked at the effects of ENSO on seasonal epidemic influenza from places such as France, the US, and Japan. And these have been done with a limited amount of data, and they're not really that consistent. Now, the problem with ENSO, and trying to associate it with the flu, to my mind at least, is that the response to ENSO is very variable. In that sense that if you look at these maps right here, what you're seeing is some areas are getting warmer, some areas are getting colder, some areas are getting wetter, some areas are getting drier. It's not consistent. So if the ENSO effect on flu is going to be mediated by changes in absolute humidity, and that's not a given, but if we were to follow that absolute humidity argument on seasonal timescales at which ENSO is impacting it, those effects are going to be highly regionally localized, okay? So you'd have one effect here where it's dry and warm, but you'd have another one here where it's wet. So if you're expecting to find some global signature that you're going to have a change in seasonal flu that you're going to be able to measure globally that's due to ENSO, you're not really going to find it based on this because you have such varied responses in different locations around the planet to ENSO. So I thought about that and I said, well, that just doesn't, this is a very hard problem, I said to myself, and I'm not so sure it's going to be really tractable. So I then said to myself, well, what about pandemic flu? And this gets to the difference between seasonal and pandemic flu. To understand this, you need to understand why you get the flu all the time again. So I referred to this before, but the reason why we get the flu all the time is that for human flu, there are 3 dominant subtypes floating around all the time. There are A, H1N1, A, H3N2, and B, okay? We're all susceptible to these, and in addition to that, these flu types go through gradual point mutations, which result in antigenic drift, okay? And these are gradual changes to the surface glycoproteins of the flu that make it so that your antibodies don't remember it. So the way your immune system works is, in a nutshell, basically the way it works is that, yeah, I'm not gonna go that far, but a basic way of the way your immune system works is it has a memory component, okay? If you're actually infected by something, your body sits there, and the first thing it does is it goes into its files and it looks through all its memory things and tries to find an antibody that previously worked to actually bind with the protein that's in there and clear the infection, okay? Bind with the antigen. If it can't find that, it then has to go through the slower process of generating a new antibody, and it's somewhat of a random process that actually will bind to the antigen and clear it out of your system. You will get infected, you'll become sick most likely, you'll shed the virus, you'll transmit to other people before you actually clear the virus. And the reason why a vaccine works is what it is it's either an attenuated or a dead virus or some portions of a virus that are injected in you. It results in a response, you develop an antibody for it, and then you stick it in that filing cabinet in your body, put it in that memory component, and if you're actually assaulted by the actual virus, you pull that out and you quickly are able to clear it preemptively before it sets up camp and you get sick and you participate in the whole transmission cycle, okay? So if it's something that's out there that's actually relatively constant, and that's not really often the case necessarily in the viral world, you're going to have that and it's going to provide you immunity for a very long time. The problem with flu is that you got 3 subtypes. So if you build an antibody to this guy, it's not going to necessarily confer cross immunity to these 2, okay? Furthermore, these guys are constantly mutating. They're constantly changing. They're reproducing so much, and they sometimes screw it up. And they have point mutations, and that results in this antigenic drift, so that you can get infected this year with H3N2. You mount an immune response. You have that antibody on record. But in 3, 5, 7 years, that virus will have shifted. It will have drifted far enough, I should say drifted far enough, that it will no longer be recognizable. That antibody will no longer be able to bind to that same antigen, and you will no longer be able to preemptively clear it from your body. So as a consequence, you're going to get reinfected with that same virus again in 3, 5, 7 years, whenever that immunity wanes, okay? That's why there's that S in the SIRS model. That duration of immunity reflects that antigenic drift time period, okay? And that loss of immunity to a specific change. So we get hit with the flu all the time because there are 3 types of it, and it's constantly changing. Now, antigenic drift needs to be contrasted with antigenic shift, okay? And this is a large or abrupt shift in those surface glycoproteins, okay? And this happens and produces something that results in a pandemic. This happens for a different reason. This happens because of something that's usually thought of as reassortment, okay? And that is that a single organism can be infected by more than one flu strain at a given time. And when that happens, particularly for the A viruses right here, what they can do is, well, a virus is just not even an organism. It's just a little bit of code. It's just a bit of DNA that gets in your body, it hijacks your cellular machinery, and it makes lots of copies of itself, which you then shed out into the environment, okay? If more than one of those viruses is in your body, it's not going to make, in a single cell within your body, it's not going to make good copies of it. It's going to start hybridizing them. Influenza is actually segmented into 8 segments, so it'll take 3 portions of one, 5 portions of another, slap them together, and you may have a very viable and radically different virus on hand because of this reassortment process. And that produces antigenic shift. That produces a virus that the world really has never seen. So instead of a portion of the population being susceptible to it, it's so radically new that 80, 90, 100% of the population is susceptible to this new little beast. And that beast can go out and infect and cause a pandemic, which is a much faster spreading epidemic, you could say, because there's such a high level of susceptibility to it, okay? So just to go over this again, antigenic drift is the small, gradual changes. It looks something like this. You get little point mutation changes on the surface glycoproteins that happen gradually over time, and that results in seasonal flu. This flu that you can get over and over again, and it's a gradual change comparatively. Reassortment is the combination of 2 or more viruses within a single cell that produces a hybrid that looks radically different, okay? That can be very different, can be infectious, can be virulent, not be virulent, and that produces a pandemic, okay? So, said to ourselves, well, antigenic shift is the key process. Rather than look regionally and try to tie this into absolute humidity effects, let's look globally, try to make it simple to begin with, okay? And reassortment is that process we need to focus on, okay? That creation has to precede the onset of a novel pandemic. In other words, when 2009 H1N1 appeared, that pandemic, at some point prior, there had to be a reassortment that took place that created that new hybrid strain, right? Let's explore ENSO conditions prior to the known documented onset of well-dated pandemics. Let's see what they look like and see if there's any consistency to it. Well, that immediately eliminates our data down to 4 data points. So, the last 4 human pandemics were 1918, 1957, 1968, 2009. Those are sufficiently well documented to allow us to say when they first emerged. You get earlier than that, you really don't know. You don't really know clearly when they took place. Well, let's look at what was going on in the Pacific. Here it is, for the conditions prior to each of those 4 onsets, and here's the onset for each of them: March 1918, February 57, July 68, and March 2009. So let's look November through January prior to it, which clears us in front of all of them. And what we see is that for all 4 of them, La Niña conditions were taking place in the Pacific. Okay, that's interesting. Is it statistically significant? That's what the patterns look like also, just to show you. Well, if we do a little bootstrapping analysis on it, we find that the likelihood that you get this magnitude of anomaly averaged is 6.9%, okay? It's not, classically at the 5% level or anything, but it's interesting, okay? Now this finding is insensitive to the precise index of SSTs that we use to describe ENSO. You can use Niño 3.4, you can use the Southern Oscillation Index. If we try to entrain the less well-documented Russian pandemic that started in 89-90, and it's a little hard to date it, but if we do that, it actually supports this as well and reinforces it. So there seems to maybe be something there, but the question is, of course, well, is this just coincidence, or is there really something causal going on there? So why might this association be real? That's the thing I ask you. Anybody got any ideas? Consultant has an idea, I know. The idea has to do with birds, okay? So birds are a big reservoir of flu, okay? Birds actually get human flu. They transmit it around. You've probably all heard of H5N1, an avian flu, something that people are worried about, that really making a big jump to humans, and we're getting a new subtype, a fourth subtype floating around that's being readily transmitted person to person. Well, ENSO has been shown to affect the health behavior of migratory birds, and migratory birds are thought to be very, very key in the distribution and movement of flu, including human influenzas throughout the planet, okay? So you have this phenomenon that's already been described to affect the health and well-being and mixing of bird species. How might that be associated with reassortment? Well, what we're hypothesizing is that La Niña conditions are bringing together divergent influenza subtypes. So you have this phenomenon that occurs every 2 to 7 years, depending on periodicity currently of ENSO, right? And what that would do is, because it's changing the local health and the support and the ecosystem resources that the birds exploit upon the flyways, their stopover times, their fitness, whether they're getting blown around by storms, whether they're actually congregating together is being changed. And so if you bring together bird species in a La Niña year that normally don't get together otherwise, those bird species have been in isolation essentially, and they've had their own flu subtypes for the last 3 to 7 years. Okay? You then bring them together and you are allowing them to have this reassortment process take place. You're bringing those divergent subtypes of flu together. You're allowing some host organism to have simultaneous infection by more than one strain of these divergent flu subtypes, and that reassortment process is potentially taking place, some of which may create a viable virus, some of which may create a viable virus that can infect humans. So that's our idea. So we're thinking that the likelihood of pandemic strain emergence through this reassortment process increases during La Niña events. That's the role for the birds. Now, if this is going to happen, then the birds have to be involved in all these processes, if this is what's really going on. Now, the 57 and 68 pandemics are thought to have originated from the reassortment of avian influenza viruses. The 2009 pandemic, you might have heard it called swine-originated influenza virus originally, was thought to have arisen from the reassortment of 2 swine influenza viruses, though there are bird components in it. Our hypothesis, if it were to be valid, would have to require avian infection and transport of those one or more of those viruses to facilitate that final pig reassortment. So in other words, birds can be infected by pig viruses as well, pig flu viruses. One of them would have to pick it up one place, fly to a region it normally doesn't, defecate. When they defecate, there's flu virus in bird poop. It could go into an area where there are animal waste lagoons, it could go to a pig farm, anywhere in these areas. It would have to introduce it into another area where there's a population that has a different type of flu viruses, and they would have to have that reassortment take place finally in the pigs. So a number of leaps have to be made there, but that said, this is a testable hypothesis, okay? Again, we have 4 data points, not a lot to hang our hat on. So how can we test it? Well, there's a lot of research that's going on bird migration patterns, changes in effective weather, so we could start monitoring it. And just use Southeast Asia as an example. Look at the changes in the bird number during the La Niña event, changes in their fitness, changes in the rates at which they're setting influenza into lakes and pools and ponds and wherever they're defecating, changes in their stopover time and what their mixing rates are. What's the diversity of bird species? Are you seeing bird species you typically didn't see in a La Niña year as opposed to other times, neutral or El Niño conditions? So we look at all that, and then the other thing to do is to start looking at viral genetic population dynamics, okay? So reassortments happen more often than the 4 pandemics that we saw there, but a lot of them are duds. A lot of them produce something that's not necessarily viable in humans. So if we can start documenting all the flu diversity that's within the birds, we can actually start labeling and finding reassortment events. We can look at the frequency of those and how that relates over time to what's been going on with ENSO to see if there is some sort of signal in there that we could back out. So these are some of the things that we could do to actually try to substantiate this and try to find it. Now, why would you want to do it? You'd want to do it because we cannot predict a pandemic at this point. We just, we follow, we grab it by the tail when it's already past us at this point. So if we had some level of predictability, if we had some notion that, in truth, when a La Niña does occur, there is an increased likelihood of a pandemic. That would actually prepare public health officials in different ways. They could start thinking about what they want to do in terms of preparedness, how they might want to increase their monitoring in those years when it is La Nina, and perhaps if we do these investigations and pinpoint certain hotspots that are critical, maybe we can find locations where we would want to look for that reassortment in particular. So, just to summarize this last bit, the onset of each of the last 4 well-dated human influenza pandemics was preceded by a La Nina conditions in the equatorial Pacific. We're hypothesizing that ENSO variability affects the fitness, the flight activity, and the intermingling of migratory bird species and alters that likelihood of host co-infection by multiple influenza strains and that consequent viral reassortment that creates a human pandemic strain. Human influenza pandemics are rare, okay? However, this hypothesis could be tested by further study of ENSO-associated changes in influenza virus population dynamics, virus prevalence in various host species, and avian migration patterns. Okay? So, conclude by just thanking my collaborators. Mel Cohn, as state epidemiologist in Oregon, actually introduced me to the Lowen et al. Paper that sort of got this whole flu gambit rolling for me. Mark Lipsitch, who's at Harvard School of Public Health, is one of my principal conspirators, you could say. And then these other folks, Virginia Pitzer, Cecile Bedeau, Brian Grenfell, and Ed Goldstein. So, thank you.
Take some questions.
I'd be happy to take questions. Yes?
Yes, Marco.
I think I have to rush off to the exam, but the question that's sort of perfectly related to what you're talking about is, I remember years ago reading an article about commercial jetliners used to release the waste from the toilets into the atmosphere as they're flying.
Okay.
You know, it would save a lot of work when they landed.
Yeah.
And a study was done, I think it was in India, that showed that the incidence of cholera in the footprint of the flight paths was significantly higher than elsewhere.
Really?
And so there was a suggestion that the aerosols, I think cholera is bacteria, isn't it? But anyway, was landing.
So there was deposition of the whole thing.
And there was so there was a pathway of infection.
Well, I can't speak to the airline toilet issue because I don't know about it. But in terms of just raw deposition of viruses, that's something that we don't know, actually. We don't know the rates at which, I mean, there are some studies now that are going on where people are just putting out petri dishes and collecting things, or active samplers. We don't know the rates at which virus is present in our environment. Because we know so little about the modes of transmission, we don't even really have a full sense of how much virus typically is out there for somebody to get infected. I mean, it really only takes one viral particle to get somebody infected potentially, but what level of it do you need in the air or on the fomites to make that happen? And is it raining out? I mean, the idea that potentially, a flu could be infected across the entire Pacific Ocean and rain out onto the west coast of the United States, for instance, that's possible. I mean, there have been some studies just recently where people have been trying to just collect it, in Taiwan in particular, and they've been finding flu virus. So is that one of the ways it actually goes around the world? And then the question, of course, is, formally if airlines were contributing to that. I don't know. I mean, it's a question.
In the modeling of the pandemic, flu, have you integrated differential lags comparing the early episodes of pandemics where travel was extremely limited and slow in transmitting disease with the newer events? So the lag to have the pandemic, the local epidemic, say, starting is differential, is different.
You're talking about the seasonal flu model?
The pandemic.
The pandemic.
Actually, the pandemic. So one can think that early 20th century to have a pandemic detected in developed countries, say, is the lag is different than later episodes in the late 20th century.
I haven't looked at it. We haven't done any dynamic modeling of the pandemic at all ourselves. But I would imagine you're right. You look at something like the Russian pandemic in the late 19th century, it took a lot longer than what you see now. I mean, the 2009 pandemic followed a pretty consistent course, and so, that would be something to look at, yeah. Do you
and another question related to the first part of the study, when you did the immunity estimates, what was the best fit model for immunity? You tested, I believe, between 2 years and 10 years.
That was the one variable that wasn't as constrained, and I think that has to do with the fact that we had combined the B and the H1N1 classes in there, and further, we don't have cross immunity in there. So because we don't have that, and because it's not very well constrained, I don't I don't think you're going to get a good constraint on it. And I was not surprised that we didn't get a good constraint on it. That was the one of the 4 variables that didn't converge to something. It seemed to be all over the place.
Okay.
Yes?
Kappa? Yeah. Go ahead.
I also have 2 questions. One is that you mentioned that there's a correlation between absolute humidity and the rate of infection of influenza. Did you do a study, or does it correlate with regional differences? Like you did a study in Arizona, which I think would have a very low absolute humidity compared to New York, which has a higher.
We didn't do a correlation. What we did was this anomaly analysis. So what we're doing is we're looking at the onset of flu, which is the anomalous event. So from season to season, flu occurs in the winter. What we want to know is when it occurs, which is the start of it, which is going to be idiosyncratic and specific to that year in question. And then we want to see whether anomalies are associated with it. That's how we looked at the data. We wanted to pull it out. If you were to just take over a long time scale and correlate absolute humidity with flu, you're going to get a good correlation, but it's somewhat meaningless. You can do it with the price of grapefruits, you can do it with temperature. Anything that's got a strong seasonal cycle, you'll get a correlation because they both have strong seasonal cycles. So we wanted to pull out the seasonal cycles from this and just look at the deviations and see whether short-term deviations that really are due with weather patterns. And the weather pattern this seems to be due to is actually a cold front coming through. So it was similarly associated with cold temperatures, low relative humidity conditions, and clear skies, a lot of sunshine. Exactly what you get after a cold front would pass through. And that's what we were seeing. Do you have a second question?
So go ahead, sorry.
Oh, sorry.
Kabazima had a question.
I just wondered why, because changes start to occur during El Niño before La Niña, in terms of the air mass flow and also precipitation patterns and stuff. Why doesn't it, the pandemic, why doesn't it occur during El Niño? It's until La Niña.
Well, there's no reason. Both of them affect conditions around the planet. One is just a different phase, and they tend to have opposite effects. The fact that it seems to have a phase preference, if this is actually true, would indicate that there is some conditions along specific flyways that are providing better resources or worse resources that's resulting in this intermingling of bird species. So the effects are not necessarily opposite. So when you have El Niño versus a La Niña, they're not completely opposite with one another, but they are somewhat out of phase with one another. So they're just flip sides of the same coin. So if you look at this right here, for instance, you can see, this one's not as good. Here's La Niña, and if you look at El Niño, you can see the patterns are somewhat opposite of each other, right? And so the dynamics, both of them are doing it. You're just increasing convection in a La Niña event over the Indonesian area that's creating a whole bunch of waves distributed in the atmosphere, creating changes in storm tracks, the way air masses affect, et cetera. So it's just as impactful as El Niño is.
Yes, Josie.
Well, in discussions with with a colleague that studies rotaviruses, hantaviruses, virologists, I don't think you don't need birds. You can just have wind patterns because they see seasonal shifts from when you see rotavirus or hantavirus in the southwest and the northeast, and it coincides with shifts in wind patterns.
Yeah, and if it's raining virus, and you have a change in the whole preferential tracks, and changes in wind patterns, changes in storm tracking, and it is coming all the way across the Pacific, you could have new viruses introduced into an area, rain down on a pig farm, and that would result in the reassortment potentially if the pigs pick it up. But I don't know. I mean, it's a question of what are the mechanisms really? Yeah, possibly.
I just want to Bob has a question.
So I remember I used to do a lot of work in the southeast United States during the summer period, and of course everyone goes indoors, air conditioning during that time. And it was obvious that people got sick during that period in the same way people in the northern regions got sick during the summer. And so I'm wondering if...
You mean during the winter? During the winter?
Yeah, excuse me, during the winter.
In the northern, but in the summer...
summer in the southeast. Yeah. And I'm wondering if the control of humidity in buildings and has altered some of the seasonality that you expect to see related to absolute humidity, and if there are optimal controls that people should seek to control their homes homes and offices and so on that would minimize these kinds of.
Well, you're touching on a lot of issues there. Well, I'll tell you. After I first published this thing on humidity, I got a lot of calls from humidifier companies telling me basically, right on, brother. And we'll be and just in terms of as a public health announcement, you know, don't go out and humidify your home willy-nilly as a response to this. Not that I'm assuming you believe it. There are a lot of pathogens that like high humidity. There's gram-negative bacterium. There are pathogenic molds that really like high humidity environment. So that's not a good course of action. The issue that you're talking about people getting sick indoors, you don't tend to see flu that much, but it can happen. It can be flu that happens. You might have rhinoviruses a lot. I get sick when I go inside in freezing conditions here because I sweat and then I come inside, it freezes on me, and I find I'm getting sick. But flu, which is a pretty severe ailment, tends to occur then. But that does not mean it cannot occur then. And it does sometimes occur in the summer. And you can have it when the pandemic took place. Given the high susceptibility, it was expected. We had a paper on this where we showed that it was expected that you would see outbreaks in the summer. And there were outbreaks aboard military vessels and in specifically, school academies, and they were taking place in the height of summer. And it was a combination of the susceptibility, getting the virus into this close environment, and the fact that it was, given the high susceptibility, you could still have transmission. And I won't go through the little bits of dynamics that are in there. If you're talking about the indoor environment, we still don't manage humidity very well. I mean, there are HVAC systems and whatnot, but you can go into these modern buildings, and I've gone in and find humidity levels where the relative humidity is 10% in the winter. I mean, it's just not being controlled at all. It's basically following the outdoor pattern, squiggle for squiggle, with every little variation that's taking place out there. And there may be other buildings where it's better, but it's something that we don't manage, and something that I think we need to think about, not just for flu, but for general health and well-being, is we've got to think about managing the humidity in addition to the way we manage the temperature. It's something that we don't do. Other thing that I'll add for you is that, one of the things people talk about, some of the other things, is post-school schedules, school calendars, people coming indoors for this behavioral response where people spend more time indoors, and hence there's more person-to-person contact, and that's why you'd see the wintertime outbreak. In the South and in the Southwest, it's the opposite. People spend a lot more time outdoors in the winter, and still they have the peak of flu in the wintertime. So it jives with humidity, but it doesn't really jive with what you would see anecdotally there, though we've done some Stuttles modeling, and it doesn't work as well modeling either. Did that answer enough of your question, at least?
How about what else?
Yeah.
My second question.
Sure.
I was born and raised in the Philippines, and it's tropical, and always my perception was that the flu season started during the rainy season.
Yeah.
So the warm, after the warm summer, the warm dry summer, and it started in the rainy season.
Yeah.
So
we actually have a paper that we just submitted, a postdoc of mine, on this, and it is associated with the rainy season. You go in the tropics. So that's what we found from looking at as much tropical data as we could get our handle on. What you'd expect for humidity, though, is if you go to a place like the Philippines. Do I have this here? I think I have it somewhere else. So here is different patterns of seasonality of flu. This is from a paper by Cecilia Boat. Here's the U.S. with this big seasonality in the wintertime. This is the winter, January, December here. Here's Argentina, temperate the other half, out of phase with us. Here's Colombia, which is very tropical, and it's pretty flat when you look at it, okay? There is a slight seasonality that seems to occur where things start in the beginning of the rainy season. If you look at this line right here, the green line here, I know you can't read the label, it says Bogota, okay? This is what humidity conditions look like on average in Bogota, which is right near the equator, pretty close. And basically what you see is that it doesn't vary much, and it's high all year round. I mean, this right here is Hong Kong, this is New York, okay? Big seasonal cycles comparatively. Bogota in the deep tropics doesn't have any sort of fluctuation. If you then take and look at this relationship right here, Bogota was right here, going like this, okay? Which gives you, if you're to believe this relationship between survival and specific humidity, it gives you almost no variation in the survival based on humidity alone. You would expect that in the tropics, you're not going to actually have a large impact of humidity on the survival simply by this relationship, if this relationship is in fact true. Whereas if you go to New York's varying on a much larger scale, and it's over here where there's a lot more bang for your buck, going between 20% and 60% 1 hour supply. So you'd expect in temperate regions a lot of modulation, and you'd expect very little by humidity in the tropics. And as a consequence of that, I think that there's got to be some other mechanism that's guiding whatever seasonalities you do see in flu once you get into the tropics. And the meteorological signal we found is precipitation.
Liu has a question.
Yes, in the very beginning you showed a very interesting similar cycle between the California and New York.
Uh-huh. Okay.
These lines are very close. If we put temperature and absolute humidity with these two curves and the single cycles of those two variables, which one will be closer to this?
Well, they're very, very highly correlated on seasonal time scale. So temperature and absolute humidity are incredibly highly correlated. So it would be almost indistinguishable. You could try it, but just putting these against here, again, you can put the price of grapefruits in, is what I always say. You could put anything with a seasonal cycle in there. It's not really giving you much information because you're not accounting for that gross seasonality that's confounding everything there.
But this also suggests that all the states in the United States will have this similar.
They all do. They all. Yeah. This is two examples.
So there are no more questions. So one reminder that for first year ITPA students, there is a sign-up sheet in the back. Please remember to sign it before you leave. So let us thank Dr. Shaman for such an interesting